The big questions I think you guys are going to try to grapple with more and more is just what is which study and why is all the studies important. This is from Harvard? Yes. We got it from Harvard ICTS. So this is from Harvard. We also have another one for you on the website. Where did we get the other one? Tufts. And so as we find these different ones, we're just going to post them for you. You're going to obviously find some similarities between them, but I think you're also going to find some differences. So I just wanted to kind of go over each of these and just answer questions as we go through them. A little bit, just so you know, the differences are going to be artificial, but it's important that, you know, we look at all different aspects of research. So, you know, T1 is really, you're trying to translate in the future, you know, it's hard to do it every time, you know, how the research is going to apply. So these are a lot of bench studies, a lot of basic science research. These are great studies. They're preclinical studies, animal studies. You know, you could look at physiology, different effects of drugs in vitro. You can look at effects of drugs on animals. These are what often are called phase 1 clinical trials, and we have a list of these at the end. And you might always hear, like, you know, phase 1 clinical trial, phase 2 clinical trial, and as they go until they kind of become FDA approved and a drug is available. Definitely these are proof of concept, which just means that somebody has a theory that something is going to work, and they want to apply it. They want to see if it actually works. And so this is kind of some of the earliest research studies. The T2 is what I think most of the time we hear about, you hear about. So it's when, you know, you're trying to then take the research and see how it applies to individuals. And that's really the T2, and it's translating it to patients. As mentioned before, you know, it says the T1 was translation to humans, and it might apply to just human physiology. It might apply to basic science principles that apply to human physiology. So T2 translating it to patients would be, you know, they test new interventions, definitely still controlled environments. You'll see a lot of double-blind studies looking at the effects of, you know, potentially one drug, versus another. And definitely important to do phase 2, you know, you're going to start looking at some of the safety stuff later on, and some of the safety stuff definitely comes in with T3 trials, which is translating it to practice, and you're going to come up with guidelines on how this is applicable in real life. And these are, again, following, so phase 2 and phase 3 clinical trials, and we'll go over those. And health services research is really a lot of the stuff we were talking about outreach. It's about disseminating what you're trying to do. So you disseminate it to the population, you tell people in the community about it, you tell other researchers about it, see how it's applicable to their research, you tell practitioners about it, and that's what, you're trying to then take some of that information and then disseminate it throughout. You're trying to see how it applies to clinical outcomes on a larger than single human basis. And that then leads to population research. So where investigators, looking at factors and how it affects, not just, it doesn't have to affect a whole group of population. It might be individuals all over the world with specific factors, or it might be a group of people in a certain area. And you're trying to look at, sometimes a drug or a treatment or an intervention might not work in a single individual, but when you look at it on a population basis, it's going to be dramatic, the effects. And, sometimes you look at the social determinants, some of it's socioeconomic factors, some of it might be, people's cultural beliefs, how that comes into effect with a region. You might have something that works perfectly for one region, you try to apply it to some other region, it just doesn't work. So for that population basis, it just doesn't work. Although the treatment works, it just doesn't work in that area. And so it's just important to realize the different determinants. We kind of put these, we're going to put this up on the website, put the source, which is from Harvard, and go through the different phase 1 through 4. We wanted to give you guys some videos that I think will give you a lot better appreciation for what's going on, and that's the current videos. These are off the Harvard Catalyst website, and we'll put this online. And just want to kind of go over the first video, have you guys watch it, and probably take it to the beginning, huh? And then once you watch it, then we will talk about it.
For the first 20 years of my career, I worked on mostly cholesterol metabolism, which is in many ways related to diabetes.
T1, sorry.
Very basic fundamental mechanisms of cholesterol transport and why cholesterol blocks your arteries. But I realized after 20 years that as exciting and rewarding as that work was, and it was, I wasn't actually producing therapies. So I took a 2-year sabbatical and I went over to work at the Novartis Institutes of Biomedical Research, a pharmaceutical company that was devoted to making therapies. And I spent 2 years in charge of their translational medicine program in diabetes and metabolic disease. One of the holy grails of drug development is to try to find targets that, based on human evidence, you're pretty confident if you can make the right drug and it hits the target in the right way, you will get the result that you want. And to do that, we've turned increasingly to look at rare genetic conditions in humans. To be specific on the diabetes research, there is a transporter in the kidney. It's a protein that takes the sugar that you would normally reabsorb from the urinary space. After you filtered your blood through the kidney, it would take that sugar and reabsorb it back into your body instead of having you have it pass through into your urine and pass out of your body. There is a genetic condition where individuals have inherited a mutation in that transporter and they can't reabsorb the sugar. We made a drug that was intended to mimic that genetic condition. We gave it to a bunch of normal volunteers. We saw the drug was well absorbed. We saw the drug lasted for a long enough time in the day so you could take it once a day, which is typically important for trying to get people to take an oral medicine. And we demonstrated that we could produce on the range of 60 to 80 grams of glucose wasting in the urine of a normal volunteer. So we were completely mimicking, as far as we could tell, the genetic disorder that led us to pick this target. So that was enormously exciting, and we hadn't yet treated a patient, just normal volunteers. So it may be that if you've never done any of this work, taking one of the several Harvard Catalyst courses about translational medicine would be a very good idea. We just created a T1 course that ran for 2 weeks this summer, focusing a lot on how you take a molecule, either small molecule or biologic molecule protein, and put it through the regulatory process to get it into the clinic and what that first trial would look for. And then there are people around the community, the faculty that taught in our T1 translational course were faculty from either inside Harvard walls, but we also invited a large number of people from local biotech companies and from big pharmas who, to a person, man and woman, said, if you have a project and you need some advice, call me or email me. I'd be happy to give you some insight. So I had to spend quite a bit of time and effort understanding drug metabolism in a way that I never had as a academic investigator, as a medical student, as a doctor. And it's pretty fundamental to being able to develop the therapeutics. Designing a clinical study that gets you an answer that's interpretable and valuable in many ways turns out to be a lot harder than certainly I think I had conceived it to be before I started doing it. So getting it really clear what you're going to accomplish in that trial, what your goal is very, very critical. You have to be passionate about wanting to take the idea and make it apply to a human disease condition, because the road is long, the road is expensive, there are many, many hurdles on the road that will make you go home and wonder whether or not you made the right decision, and then understanding what are the hurdles and finding the resources, both intellectual and financial, that's going to enable that to happen are the two critical pieces, in my view, about making a decision to go do this if you decide you want to do it.
So I think one of the questions you guys might have, and I just want to kind of bring it up early, is, what things could you anticipate, and I'm not a very test-focused person, but on tests, and I think, being able to differentiate the different I will definitely, one way to, I think, look at this is give you examples and for you to be able to demonstrate them, or we'll ask you to give us an example of one. So I think that's one way that we would ask you, just an important concept based on today's, what we're going over. So the presentations that you guys do at the beginning are not something we're going to test the group on. These are for the individuals involved and for you guys to use this as kind of fuel for thought as you're trying to prepare for your own presentations. And I hope just the fact of presenting will help you in the future if you don't get enough of that experience already in your different classes. So the part that we're really going to try to focus on, questions for if you're at all wondering for the exam, and we'll talk more as the weeks go on, are going to be probably the presentation that goes on, the 1-hour presentation. We'll try to give you good resources like the Harvard Catalyst program. We're going to go over the different videos, and you can use those as some material to study. We'll give you some examples of what T1, T2, T3, T4 is, and that would be today's focus, and also next week's focus as well. The lecture by Dr. Cooper, I believe, is coming next week. So what, I know he covered a lot of different things in his presentation, but what is T1 research, that he covered? So he kind of jumped around it, but I just want to make sure that you guys realize what is T1, the specific T1 that he was talking about. Yes?
He was studying cholesterol and how it affects the arteries, I believe.
Well, he was studying quite a bit of things, but I think the part that I just want to point out that was most effective is he was saying, how does the physiology of the person get affected by the medication? So first, he's trying to come up with a concept. So T1 is kind of the ground of where you're trying to come up with a research study, right? So what he was saying is, you got all these different thoughts going on. You got all these things that you're trying to help, but how do you get started? And he was demonstrating for you, and we'll give you the website so you can look at this again, but he was demonstrating how difficult it is to try to get your hands around it. So one of the topics he was talking about was diabetes, for example. And he was talking about, wasting of sugar, glucose in the urine, and how do you work at reabsorbing that glucose? And he was saying, well, you first have to understand what is happening in the body, in the human body. What is the issue? What is the problem? And then figure out, what's the best way, now that you know the problem. So you first come up with a problem, and then what are potential ways that you can address it? Are there, is it based on medications? Is it based on a defect in the human, genetic process? What's the defect? What is the genetic defect that's causing that? So before you can even design a study, what he's pointing out, and he kind of goes around all different topics trying to point this out, is you first have to really study it. And I think that's why T1 research is so difficult. I think that's probably, and my brother's a basic science researcher, so my hat's off to him, because he does these T1 research in vitro, in labs, on mice, very basic principles of mechanistic research. He does this research. And I just want to point out through this video that he's trying to grapple the issue of how do you even approach it? And you have to approach it by looking at the physiology. You have to approach it by looking at potential genetic defects. You have to approach it by saying, how do we even study this? What's the base? Because when you go to T2, when you go to T3, when you go to T4, you're kind of basing what you're doing based on previous research that's been done a lot of times. And, I'm not saying it's easier or harder, but it's just different. In T1, they're kind of de novo trying to create principles. They might look at a clinical quandary, but then they really have to break it down into the different pieces. And without breaking it down into all its different components, you're never going to be able to solve it all by itself by just saying, well, I'm just going to try this drug and see if it works. It's never going to work. So first you need to find out, well, what is the problem? Why is it a problem? Why is it even important to study? What are the clinical outcomes that are creating this an important issue to resolve? And then look at and see, is there a mechanistic way that I can understand what is going on? And is there a drug or a treatment or a genetic defect that we can identify that's causing it? What's the best way to address that defect? And that's what he was trying to point out. And as we go through, we're just going to study diabetes in the 4 examples today, so hopefully that will give you a little better perception. Yes?
I was just going to point out, they say that T1 is called the translation to human aspect. And so he also mentioned that one of the things that exemplifies T1 is this first in human concept. So he said they used healthy volunteers first. And that's also something that happens with T1.
Okay. That's good. So hopefully that gives you a little bit of a perspective, and now we're going to look at T2 and the same thing. So it's a little bit of a different study.
So I started in the bench, really doing basic science research, and wanted to work more directly with people, and therefore taking the ideas that were closest to the bench and asking how can these be made applicable. I work very closely with several basic scientists here at the Johnson Diabetes Center who have been studying mechanisms of obesity-mediated disease and how that may link to the development of type 2 diabetes. They discovered that certain pathways were involved that included activation of a chronic subacute low-grade inflammation, and suggested that anti-inflammatory drugs may actually have a potential role to treat or prevent diabetes. We moved these into the clinical arena first in very small phase 2 clinical studies, which were proof of concept, showing that we could indeed improve blood sugar over the short term, and then moved into multi-center trials that have been ongoing now to try to see whether or not these drugs that have been approved to treat arthritis as their fundamental original indication may actually be useful to treat patients with type 2 diabetes as well. We actually just reported our first phase 2/3 multi-center trial, which was a 3-month study looking at whether the drug salicylate can actually lower blood sugars in patients with diabetes. It was a dose-ranging 3-month investigation, and we were very pleased to find that it lowered fasting blood sugars and hemoglobin A1C, a measure of average blood sugars in patients with diabetes, where we're taking an existing compound and trying to ask if there's a new indication for it. So we actually don't need to go through the first in humans or the pharmacokinetic profiling of the drug and move in first to ask the question, does it improve a measure of blood sugar or insulin secretion or insulin action in a population that we think it's appropriate for? And then from there, one can develop trials to actually ask whether it leads to improved health outcomes. The product already has a long-term safety profile in people, is inexpensive to manufacture and generic. Therefore, the potential cost-benefit ratios, if it's effective, might be substantial. So I think that my research goes well beyond testing this particular drug. And I think that the strengths of what we do really take things all the way from proof of concept into people, all the way into multi-center phase 3 clinical trials quite independently. We also have tremendous collaborative efforts to try to make the original discoveries, whether they start in the laboratory, whether they start within observations from our patient population, and move these forward by going back and forth between the bench and the bedside. It's only through research that we will come to new treatments and cures for our patients. And it's only through the better understanding of what's going on within our patients that can be best carried forward in the laboratory. But what's found in the laboratory is only relevant if it's really relevant to people. The obstacles are pretty straightforward when you do clinical research. The first is to get the funding. The second is to generate the protocol and get the IRB approval to do the work. The next obstacle is to actually find the patients who are appropriate to be studied, and then to keep them within the study for the duration. And then the fun part comes where you get to analyze the data. I actually should take that back. The whole process is fun.
All right, so hopefully that gave you a different perspective of T2 studies. So so I just want to just go over, whenever terms come up, just want to make sure you guys know a multi-center. So what's the difference between a single center and a multi-center? It kind of seems obvious, but. So you're trying to get multiple different sites. But the more important thing about multi-center is you're trying to find the patient characteristics and the numbers of those patient characteristics that you would not be able to find at a single center. So, it's a natural way to make sure that you're getting, varied types of patients. You're trying to get not just that it works in your specific patient population at your hospital, but, for example, I look at screening and brief intervention for alcohol. We have a trauma center and we have a hospital here, but that by itself is not sufficient. We want to look. We have another center that we have. It's Mission Hospital, South County, different patient population. We have Kern County. That's middle of California. We're also enrolling at Stanford up in Northern California. These are all universities, so we're also looking at enrolling patients at community clinics. We have our outreach clinic, our UCI outreach clinic. We're looking at another site which is in Puerto Rico. So you're trying to get the variety that would best represent what you're trying to address. So they had two specific things. It was very interesting. So diabetes is obviously a really important topic because it affects so many people. And I think from all of the chronic diseases, it's one of the ones that will always come up in the future for you. You'll hear about it, and it's one that people are worrying the most about, or one of the things that people are worrying with, increasing childhood obesity in the United States, because that, and all over the world, that might create just a conundrum in the future if the rates of diabetes go up, diabetes type 2. And so what she was mentioning was, she mentioned diabetes was the disease they were trying to target, but they were using a treatment that was already in effect. And what was that treatment? What were they trying to use? I don't know if she mentioned the name of the drug, but she just mentioned it was a drug treatment.
She used arthritis pills.
Okay. Yeah, anti-inflammatories. I'm assuming they were non-steroidal anti-inflammatory, but she didn't say what they were. She just said arthritis medication. So she said, we already know the medication is safe. We already know the side effects of the medication. We already know what it does. So we're not using a drug that's never been used, but we're trying to apply it in a different area. And, kind of show proof of concept that it works in this situation. So I thought that was very novel. But she already based it on previous research. So her previous research that she said is there was evidence, she didn't go into specifics, that maybe there's an inflammatory component to diabetes. So she already had that knowledge from previously, and so maybe we can try something to address that inflammatory component. And that's what they were hoping to address. And I think that was beautiful, looking at all of the effects. And she kind of went through, all the different parts of a study. You need funding for it. You need to get the patients. You need to have the, patients stay in. You need to follow them up. You need to be able to even enroll the patients. I don't know if she mentioned that part, which is some of the hardest parts of it, which is, getting the protocol, IRB approval, making sure that you address all the concerns and patient safety concerns, which are sometimes the hardest. Then enrolling is a whole nother thing, and then following up, writing it up, analyzing, all that stuff. But I do agree with her. I think, the whole process is amazing to me. And number one, it works. Number two, we can come up with new methodologies. And then, a lot of times, and I don't know if you guys have heard this, but when a potential treatment is only studied in specific circumstances or not on large enough populations, then when they keep studying it some more, then they figure out, wow, it might not work as good as we thought it did. And it kind of goes out of style. And then sometimes it might come back for a different disease or different treatment. So anyway. All right, so that was T2. And just any questions on T2? All right, let's see. And interestingly, as the they only have one example on T3 and T4, which I thought was interesting.
Type 2 diabetes is an increasing epidemic, and the striking thing about it is that it's very preventable with lifestyle changes. And the Diabetes Prevention Project is this great study that showed if you lose weight and you diet and exercise, your risk of developing diabetes drops by 68%. I'm a primary care physician by training, and so I bring that perspective to this work that I do. And I'm struck by the need to motivate patients who are at risk for diabetes to change their behavior. And so the study that I'm working on is to ask two separate questions, which is, does the result of a high genetic test risk for diabetes motivate you to adopt lifestyle changes? And conversely, because we don't know what the test result is until we actually do it, would getting a low test result make you less motivated to want to participate? It's a randomized clinical trial where we take patients who are at risk for developing type 2 diabetes, and we randomize them to get genetic testing or not. And then once they've been randomized, the patients will then participate in a 12-week weekly group session patterned after the diabetes prevention program to lose weight and to exercise. Prior to participating, we do a genetic counseling session, and we explain to them the details of the result and show them how their relative risk has changed. And now we're waiting to see whether they're more likely to participate and whether they're more motivated to change their behavior. Most genetic counseling is to provide information and let the patient decide what to do. But here, the genetic counselor is both giving the information and also using that information to encourage behavior change. The other thing that's been interesting about our study, or innovative about our study, is that we're having the data, the blood drawn, and the DNA genotype at the Broad Institute. This is their only study where they're actually, the data is coming back and being fed back to patients. This study is a pilot study, and our outcomes are surrogate outcomes. We're looking at attendance at all 12 sessions. We're looking at change in surveys about motivation from beginning to end. If we get a positive signal that genetic testing actually makes them more likely to attend and more likely to feel more motivated to change, regardless of getting a high or low result, then the next step would be a larger study and actually looking to see if we can prevent diabetes. So a lot of organizational obstacles, figure out how to explain to patients what a genotype score means, how to put the idea of 34 risk alleles into words that are meaningful to patients. So we had to develop counseling materials. We ended up using the idea of a scale where before you're genetically tested, having metabolic syndrome, you're in a group of people with a certain risk, and then by getting your genetic test, the scale can move you up or move you down. That was the visual image that we settled on. And then also coordinating with the Broad to get data back in a useful way and how to present it. Those have been the main challenges. Well, the resources you need for T3 research, I mean, it's really a broad range. You need to have a very competent research coordinator and coursework in how to organize a database for patient recruitment and enrollment. One real key of T3 research is to be able to identify patients that you want to enroll. And then you can't just go out and talk to those patients. You need to work through the primary care physicians who care for those patients. And so it's important to have good relationships with the primary care doctors whose patients you want to enroll in your studies. A big part of effective T3 research is not just to take the new scientific evidence and put it into practice, but to take practice and work backwards and ask yourself, how do patients really interpret new information and what actually is going to make change? And so it's important to really understand what the true barriers are to effective care before you try to implement new ideas.
So I really like this one. I don't know what you guys think, because mainly it kind of gives you a little kind of overall perspective of what's going on. And it talked about a lot of the barriers. And I think one thing you'll start realizing once you start rotating and being involved in more clinical research is there are a lot of barriers. You know, we, because of patient safety, because of, you know, how we are, you can't just go up to people and do all kinds of stuff. And so it's both good and bad, but they were talking about, you know, working with primary care doctors. He was talking about, you know, how you interact with the patients. He was talking about, you know, how you approach the whole questions. And he was saying all of those barriers go into the solutions you're trying to find. And you have to make sure the solution you're trying to find meets, or at least can work through those barriers. What did you guys take away from this? I just want to see what you guys thought on this video. And how does that affect what you think of T3 research? So he's putting genetic counseling together with lifestyle changes together to see, if people are more open to it. I think, one thing you're going to realize a lot in clinical research is that even though a treatment might work, it's not going to work for those people. And what he was highlighting was that we can give the patients and the individuals, we can give ourselves all kinds of recommendations, but that doesn't mean it's going to happen. So he was trying to show them, are they at risk of having diabetes? And going through a several week program of showing that, and then trying to show that if you did these things, you can then modify your risk. And actually, let me help you modify that risk. And by the end, then say, you've just modified it by this much. And now are you willing to, continue doing that? And I thought that was remarkable. Although, I don't know, maybe it seems simplistic to you guys. This is what kind of changes, people for the longer term. If you can internalize something that you're doing, behavior, if you can see the importance of why you're making that change, if you can actually go through with them and help them make that change, there's a chance that that's going to stick and it's going to modify them. Because, the best thing anytime you can ever do for a person is prevent them from having a condition. You prevent them from having that condition, it's better than any treatment you can give them after they have that condition. And I think that's one of the things that I think we need to do a lot better job in the United States is preventive health care in every way. And, people might argue that it's also tied into access to care. It's also tied into, different localities in the United States. But I think a lot of it has to do also with the importance of preventive health care. And I think that might be one of the things that we're looking at a lot more with the new healthcare changes in the U.S. is really giving a lot more importance to preventive care and primary care. And what can you do because, as you know, our costs for healthcare are very high, but we're offering that cost at the very end of the disease, rather than either at the beginning of the disease or preventing it, or minimizing the effects of the disease. And that potentially is one of the best areas. And so anyway, I thought that's pointed that out to me. So just now the last one, see what you think of this one. Yes?
So he did all the diabetes testing. Do he have to go through T1 and T2? Or if someone have done those research, he could just use those and do T3?
Yeah, so he's using previous evidence. So he's using what he knows, that you can genetically test for it. He already knows that you can genetically test for it. He already knows that he was saying based on, I think, a cholesterol or based on another treatment plan, this 12-week plan of intervention works with people. It had nothing to do with what he was trying to do with it. It was a completely different way. He was saying, we used this model of how people went out to individuals, educated them, tried to modify their behavior, and he was applying it to this specific treatment. So he was using other models, so you don't have to go through every step as you're going. Number one, he wasn't doing a drug trial, which is a whole different thing. This is a behavioral trial, and sometimes some of the more effective ways of intervening in people is behavioral and potentially non-medication related.
Do most T3 studies kind of look at how the information is presented to the patients through which mechanism? For example, the model. If the model looks confusing or if someone doesn't really quite understand the genetic testing, I know you touched upon that as well. Are there any T3 studies that kind of compare the use of different types of models to see what is more effective in changing behavior?
Well, I think it might, absolutely. Obviously, he was doing a really small project, I think, with very small number. I think if you were doing a multi-center site with much larger, so this would be, he could do a single site to see if it works. It seemed to me that they hadn't really tried this many other places. And if it works, and then if he wants to expand it, then comparing it to other models. But I don't think, a lot of times with these behavioral interventions, you could try all different models, but most of them kind of work. So it's just what fits your population and your disease and the way you're presenting the information. So what I'm saying is, he was following a 12-week model. He was following the way that he was graphically showing it to individuals. He was involving, different people to provide the educational information to the folks. So I think that's obviously a very intensive in-person way. Maybe other ways of doing this is through electronic mediums, not in person, so that might be a different model. Another way might be educating the primary care doctors and see if they can then make a difference. So instead of you trying to do it with this very resource-intensive way, so you could do all these different ones, but they might be different research studies all to themselves. And then, it's a lot easier to study one drug against another. It's much more difficult to study some of these interventions against another. And so what you end up looking at is you have one intervention, one behavioral intervention, and then you follow them up in 3 or 6 months and see their effects. So see their long-term effects. Then you have another one and you follow them up. And whichever one seems to address your local resources in your medium might be what you end up using. And I think this is our last video. And this is our T4 implications for population health.
The electronic medical record as a translational research tool is really exciting. It has tremendous potential, particularly as nationally we're moving towards having a much more cohesive electronic medical record system, as you know. And in addition to patient care, which is really obviously the primary goal, there is public health surveillance and also electronic medical record-based clinical research. My specific research fits into the translational continuum at the sort of T4 end, which is at the population level. It's a collaborative effort between our department, the Department of Population Medicine, and our informatics group, and collaboration with the Channing, and then with the Children's Hospital Informatics Group. And they're together part of the CDC Center of Excellence in Bioinformatics. I helped to write the algorithms that capture, through the electronic medical record, patients that have those conditions. And you might say to me, well, why not just look at diagnosis codes? But diagnosis codes actually miss a lot of people for a variety of reasons. So what we do is we use lab data, demographics, sort of blood pressure, weight, and medications, and we put them all together and develop these algorithms. And it's actually much more nuanced and complicated than I would have thought it would have been, starting in. But once the algorithms are working, then we implement them, and the information is transmitted to the Massachusetts Department of Public Health. The Department of Public Health, or the CDC, can stratify and ask, in this geographic location, how many people of this gender and this ethnicity have this type of diabetes? How is it being managed? What are the complications? Are the complications being managed according to various guidelines? And then a second step, once all of that is in place, is to use the algorithms to feed back to the T3 level, at the clinic level, to sort of inform interventions to improve diabetes care at the level of either the patient, the provider, or the practice. I would give, I think, 3 levels of advice to someone wanting to do T4 research. One is, definitely having a strong background in biostatistics and managing, understanding the research methods needed for looking at large numbers. The other thing I would say is take advantage, especially if you're in this area, of the remarkable T4 level work that's being done. My last bit of advice
would be to collaborate with others and really open your mind to those collaborations. They don't necessarily need to be within the same disease type, but with my experience from this and collaborating with, informaticians and just the different people involved in this large project, reaching out to investigators that have other skills, whether it's economics, whether it's health policy, and drawing it in. And along that line, keeping your mind open to the other T levels. I think early on in a research career, finding a good mentor is a critical step to take if one can. So think hard, what are your strengths and weaknesses in terms of communicating, getting jobs done, to what do I need out of this and want out of this relationship. Because it really isn't just having someone who has the hottest research topic. It really is a relationship, at least in my experience, that defines how you view science and how you ask questions and how you answer them. And is a role model, in my own experience, for creative collaboration. I think the efforts that the Catalyst is making to have investigators be aware of each other's work, stimulate dialogue and collaborations that might not otherwise have happened is really exciting. And at the junior investigator level, with some of the grants and support that you have for junior investigators, I also think that that will be very helpful for them.
So as, just to kind of go over a little bit, so this electronic medical record, you might have heard, and you might already where you are getting your health care, they might have electronic medical record. So it's similar to an electronic health record. An electronic health record includes all kinds of electronic medical records. So they're kind of synonymous, but also a little different. So an electronic health record is putting all kinds of information, both the information you put in, the information the doctors put in, the information your lab tests, your x-rays, all of it come together. And what it allows individuals to do now, and although we're kind of behind the times a little bit in the US compared to some countries in the world, is then do a lot of population-based research studies. So, you could, for example, say that, well, I'm thinking about doing this specific study at UCI. But it's a study, I want to look at what's already happened. I'd like to know, all patients with type 2 diabetes that are between, 20 and 25 years old. Well, with an electronic health record, you're able to pull that data very quickly. And if you don't have the sufficient numbers in the past, you're not even going to think about doing the research study at UCI. You're going to say, well, it's not going to be possible. I'm wasting my time. I need to do it, at multiple centers. I need to invite other hospitals. But even then, you'd be asking the same question. And some of the things that are really now being looked at are the associations between disease, the outcomes from hospitalization, the outcomes from different treatments in the hospital, and the electronic medical record is hopefully really going to transform that, and that's why the government in the United States has made it a requirement that all hospitals, all clinics will need to be moving toward this. And the hospitals, this is a humongous undertaking for them, and I'm glad that's happening. I think what it's going to potentially improve is your access to your records. It should improve that, but also hopefully improve if you're able to access your record, then you can share that with other people that need to have access, like the consultants, other doctors. So because of all the patient privacy rights in the United States, people can't just share your information with one another. You have to be integrally involved, which is, I think, pretty important. But when you're looking at a population basis, that might not necessarily be the case, because if you're not pulling identifiable information, you're just pulling whole hospital information, that becomes a lot easier. And that's what the electronic health record is providing for us. And that's one of the biggest movements here at UCI is the whole hospital at every level. If you're ever volunteering here or other places, you're going to see that people are moving to more electronic mediums, typing stuff in, dictating stuff in, whatever it is, and looking at, electronic x-rays, looking at electronic labs instead of printed information, and, making sure all of these communicate with each other. You know, one of the important things she mentioned about a little bit the qualifications for doing population type research, and I can definitely tell you, you know, Dr. Ogen-Seitan was mentioning, you know, a public health program really lends itself to those kind of research studies, because you get the epidemiology background, you get the biostatistics background, you get the whole population factors that go in, and it kind of gives you some perspective of why it's important to do that research and how you might do that. And then it also gives you a lot of the community type interventions that how you would then take it out to the community. So, you know, part of, I think, T4 research is not just showing the effect in a population, but, you know, would it actually change the health in that population and how it would do that. So all of these are available online. These videos will provide you the link. And in addition to the videos, they also have further information on them that kind of spells them out about, you know, further information that's not in the video itself. I'm going to give you guys how long do we have a break before we come back? 15 minutes?
10 or 15.
Okay, 10 or 15 minutes. How about 15 minutes? 10 minutes, and then we'll answer questions. There's a restroom out here. I think there's restrooms across the hall as well. Yeah, and there's a, you know, coffee place down on the first floor if you want to grab some caffeine. And any other questions, please come and let us know.